Anyway, my name is Claudia Traa. I am mom to James Ross Traa, also sometimes known as Risa. Um, and he is 29 years old, and um, he has two sisters, and they're both younger than him, but they're very involved with the registry and the sibling group, and are so thankful to have chromosome 18 registry for them as well. And my husband Jim is always very involved and is very appreciative of all that the registry does for all the families. I've been involved since I moved to San Antonio, not on purpose, but very happily, where the registry is headquartered back in 1997. And then in 1999, I started, or 1998 and 1999, I started volunteering with the registry and helping out. And then I did my first conference. I chaired it, and it was in 1999. And I soon became the executive director for chromosome 18. Now I had a small respite in between being executive director and just a parent, which is always my favorite role. And now I am on the board as the vice president of development. So that's me and who I am. And I just want to go ahead and what I'd like to do is say how very excited we are to have all the families all together virtually on this very special World Chromosome 18 Day. We're thanking you for joining, and we're going to have a day filled with connection and education. And I guess one bit of housekeeping maybe that Neil meant for me to share was, please be mindful that we're recording, and we're also, we all have mute buttons. And so when you're not speaking, it's helpful so you don't get background noise to keep it on mute. So just be mindful that we're live on Zoom. So be careful of all those things. Yeah, so I'm just going to get started by I want to give you some facts about chromosome 18. Um, Chromosome 18 was founded on April 6th, 1990, over 34 years ago, by Dr. Jannine Cody after her daughter Elizabeth was born. I'm sure most of you know both Jannine and Elizabeth, and if you don't, you need to. So anyway, we're so thankful for them. And Chromosome 18 Clinical Research Center is the only clinical research center in the world dedicated to chromosome 18, and that's right here in San Antonio, Texas. This is our third annual World Chromosome 18 Day, and it's our biggest virtual event of the year. From our last survey, 99% of our members reported life has been affected in a positive way by the registry. And I think it's saying in a positive way is putting it very mildly. For the research center, we have over 730 people participating in chromosome 18 research study. As far as new members, last year we had 190 new members join. The registry has approximately 4,500 families in the US and 2,000 around the world. And I know I love when Jannine tells the story about how when she wanted to start some kind of a group, they were like, oh, there's probably 40 in the world. And she found all, 2,000 of them at that time. So anyway, so thank you again to her. Uh, the registry has raised nearly $6,000 to help support our chromosome 18 families. That was last year, $600,000. Chromosome 18 board consists of 12 volunteer directors. And please reach out to us if you are interested in joining, because it's not a clique. It's all of us. This is our organization, and we want you to be a part of it just as soon as you feel you are ready to do that. And it's a wonderful group. Let's see here. World Chromosome 18 Day and other events are free to our families. And thank you for your donations, because that's what makes all of this possible. Let's see, we couldn't continue our work without, this is very, very important, without our year-end giving. We really, really depend on a big boost in December. It's the end of the year. It's very important for our programs. It's very important for our financials. That if you can, even if you did already, if you can give something and help out with our end of the year giving, that would be very much appreciated. When you're making your donations in December, just please keep in mind chromosome 18, and every little bit helps. And please get involved with the Phantom tea and the Phantom wine if you can. It's such an easy way to get other people informed about the registry and get them involved with helping this organization, which is so very important. I want to take this time to introduce Matt Miller, who many of you know. He in 2023 hosted in Columbus, Ohio, our Chromosome 18 conference. I just got distracted by something that popped up. Yes, he did. He and his wife, and he has a daughter, Macy, and Julie Miller, and so they all hosted a conference and did a great job. And I wanted to introduce him right now. And Matt is on the board and is going to take over now for me. And I just want to say thank you for letting me say hi to everyone and welcome, and always know that you can always reach out to any of us for any questions you might have. And again, consider being on our board. Matt?
Thank you, Claudia. And thanks for sharing all the information with us. I think that's always good to go over, and for people to have that opportunity to hear those pieces of information. Just real quick, for those of you that are on here, I know we're maybe running a little bit ahead of schedule here, so that's good news. But I had the opportunity to, or I took the opportunity to sit through the European call and the Canadian call today. And I was mentioning to a couple of them that the great thing that runs through this, and we had, I think, Scotland, Ireland, Germany, the United States, Canada, all on a call at the same time, which is really a neat thing. And what I saw was, and what I heard was, the need to connect and people connecting to each other. And that goes across every country line, that people need to feel connected to others who understand what's going on in their lives. And I think that's one of the great things that Chromosome 18 does. So my shameless plug is I do have a site out there for the wine and the Phantom wine and tea. And I will let you know that Julie and I have decided that we will match up to $1,000 in donations. So if anybody donates, Julie and I will match up to $1,000. So we're hoping that generates a few extra giving dollars this year for Christmas and the beginning of the new year. With that being said, I would be amiss, or we would be amiss here, if I didn't thank Gordon Hartman Family Foundation for sponsoring this World Chromosome 18 Day. They pledged a few dollars to make this happen, and we're so grateful for their support, and for those of you that are supporting us as well. With that being said, I'm going to make an introduction for somebody who needs no introduction and has been doing great work for us from day one. And really, without this individual, we wouldn't be sitting here today having these discussions. So the introduction is for Dr. Jannine Cody, and I have been fortunate to get to know Dr. Cody a little bit more here recently in our board meeting that we went to, or I flew down to San Antonio, and Jannine and I got to have a little bit of a conversation as we were running around getting to different events. So I got to know Jannine a little bit better during that time. But in 1990, Jannine founded the Chromosome 18 Registry and Research Society as a way to bring affected families together and to learn from each other. And again, that piece to learn from each other and to connect is just such a huge part of this. To date, the registry includes more than 6,000 families affected by chromosome 18 abnormalities from around the world. In 1991, Jannine enrolled in a PhD program at the University of Texas Health Science Center. Go UT, I see they won yesterday. Uh, not so much for Ohio State. I felt like it was a bad day for me. But UT, good job. And she graduated in 1997. Jannine is now a professor in the Department of Pediatrics at the UT Health Science Center at San Antonio. So with that, we're going to go ahead and kick off our day with 2 research updates with Dr. Jannine Cody, and then a Q&A answer to follow afterwards. So I will turn it over to you all.
All right, thank you, Matt. If I can add one thing to your introduction, the Gordon Hartman Family Foundation is actually the foundation that supports Morgan's Wonderland. So you may not have heard of Gordon Hartman, but I think all of you have heard of Morgan's Wonderland that's right here in San Antonio. So that's our sort of connection, or their connection to our world. So thank you, Matt. And let's see. First, feel free to ask questions at any time. If you put something in the chat, I won't see it. I'm going to depend on somebody else to tell me what's in the chat. And hopefully I'll finish in time for questions, because I felt like this time I could take a little more time than at the conference to hopefully provide a little more in-depth explanation. So let me share my screen. And I think when I do that, then I can't see any of you anymore, unfortunately, because I don't know how all this works. Let's see, here we go. Here's my screen and slideshow. That didn't, come on, there we go. Okay. Oh, I can still see a few people off to the side. Okay. Welcome to World Chromosome 18 Day. I'm excited to be able to talk to you all today. And whoops, to my disclaimer that I have no actual or potential conflicts of interest in relation to this presentation, and I'm not going to discuss any unapproved investigative use of any commercial product or device, and neither will Dr. Hale, Gelfand, or Karkar when they present later today. And I am, as Claudia said, an employee of University of Texas Health San Antonio. Whoops, I touched something wrong there. Okay. And today we're going to talk about an overview of the conditions, especially because this is a worldwide event. And so hopefully there are people who haven't heard this 30 times at the conference and are new to this. So I want to give those people some background. And later I'll talk about, in my second talk, our strategy for treatment development. And then Dr. Gelfand will talk about current projects. And so the overview of the conditions, I think as everyone on this call understands, we group all the chromosome 18 conditions into 5 different groups. But just to orient you a little bit to where all this comes from, as a reminder, here is a picture of chromosomes from a blood cell, specially stained and viewed under a microscope, and then organized. They don't look like this in the cell. They're organized in pairs, and they're organized from largest to smallest. Largest is called number 1. The smallest really is 22, but there's 23 pairs. So the 23rd pair, in this case, is an XY. This is a male's chromosome analysis. If it was a female, you would know that because there would be 2 X's and no Y there. So these are the 23 pairs of chromosomes. 1 from mom, 1 from dad. And you will notice somewhere on each one of these is a constriction, a waistband, and it's called a centromere. And that divides the chromosome into two parts or two arms, a long arm and a short arm, because the centromere is never exactly in the middle. And the convention is that the short arm is called the P arm. I think it was for petite. You can think of it that way. The long arm is called the Q arm for no apparent reason. It's the opposite end on the keyboard, same row. Who knows? Next letter in the alphabet, your guess is as good as mine. But this is a convention we'll be using, and we'll talk about the P arm or the Q arm. So just to orient you to that. However, this is a slide some of you have seen many, many times. Anything you could do to this picture with scissors, glue, and a copy machine could really happen, and it does really happen. And it happens much more often than you would ever think. So this is a more recent study than what I usually have been talking about for the last 30 years. And if you look at all conceptions, 30% of those don't even implant. Almost all of those have some sort of chromosome abnormality that make them completely unviable, and they don't implant in the uterus. And another 30%, so 60%, are lost so early in pregnancy, a woman doesn't even know she's pregnant. So that's 60% of conceptions right there. Almost all of those have chromosome abnormalities. And once someone knows they're pregnant, there's 10% of those babies are lost in a miscarriage and are aborted naturally. And most of those, especially those early on, have chromosome abnormalities. And so that only leaves 30% of all conceptions that result in a live birth. So the point of this is chromosome abnormalities happen all the time. It's just a very small number are survivable to birth. And in fact, of that 30% that survive, only a half of 1% of those actually have a survivable chromosome abnormality. So it's really a very small number. But the point of this is this happens all the time. It's a normal part of biology. We were just lucky enough to have kids that survived. And oops, I want to back up a second. And there are no data supporting any evidence that it's more likely to happen due to exposure to something. So it just happens. So to introduce you individually to each of these conditions, first we have trisomy 18. It's by far the most common of the chromosome 18 abnormalities. It's second most common after Down syndrome. 90% of these babies, and now it's even fewer percent in recent years, survive to see their first birthday. Much more intervention is beginning to happen and be accepted in the medical community. So more of them are having heart surgeries and the other interventions that help them survive longer. And as far as the research goes, well, and the registry really, individuals with full trisomy 18 tend to be very medically fragile, which is really unique compared to all the rest of the chromosome 18 abnormalities. So most of the people with trisomy 18 who are involved in the chromosome 18 registry have different versions of trisomy 18, of which there are a lot, and I will introduce you to those. So trisomy 18 is 3 copies of chromosome 18 instead of 2. So here we have, you can see here's full trisomy 18, but someone could be mosaic for trisomy 18. So some of the cells in their body have trisomy 18, but some of them are completely normal and they have 2 copies. And the number and proportion of each can vary. And it can vary and a lot. And the only real way to know is a chromosome study where you look at them under a microscope. So here's an illustration where each one of these hexagons represents a normal cell that you're looking at under a microscope. And these 5 have trisomy 18, and all of these are normal. So there's 25 normal chromosomes and 5 with trisomy 18. So 5 of the 30 or 17% are mosaic for trisomy 18. However, that was just in that blood sample that day. We have one participant who is mosaic for a different chromosome 18 abnormality, but excuse me, in their original blood analysis, they had 19% mosaicism for their condition. In one skin sample, they had 68% that had the chromosome 18 abnormality, and in another blood skin sample, 94%. So even different tissues were different, but even the same tissue, 2 different pieces of skin had drastically different levels of mosaicism. So in general, you might assume that someone who is mosaic is going to be more mildly affected because they have normal cells, but there's like no way to know what organ or part of an organ has what level of mosaicism. So going searching for the exact number isn't worthwhile, but it is worthwhile to know. In fact, they are mosaic, and they probably are less seriously affected than someone who's not mosaic. And that's just one variant of trisomy 18. There are others. So individuals can have trisomy 18 in which the 3rd copy of a chromosome has a deletion, here of 18q and here of 18p. So this is partial trisomy 18. So the 3rd copy of 18 isn't a full, complete copy. And they can also have their 3rd extra chromosome be a ring. And so this can be really confusing for families because they get told their child has ring 18, but they seem more like a trisomy 18 child than a ring 18 child. And that's because it's really the 3rd chromosome that's the ring. So this is also really a partial trisomy 18. So lots of variation, but there's more. Someone could also have 2 perfectly normal copies of chromosome 18 with an extra piece, in this case of 18p, stuck somewhere else on any other chromosome. In fact, it also could be on chromosome 18. So although the registry doesn't make this distinction, at the research center we do, and we call this 18p plus, because there's still only 2 copies of chromosome 18. And it's the normal number of copies of chromosomes. It's just that one of them has an extra piece, and that extra piece is 18p. Likewise, you could have an extra piece of 18q stuck anywhere on the chromosome. So again, it's exactly the same. There's 2 copies of 18, and the total number of chromosomes is the same as normal, but there's an extra piece of 18 stuck on somewhere else. Could be on 18, could be anywhere else. Lots of variability in the trisomy 18 group. And as I said, anything you can do with this picture as scissors, glue, and a copy machine can really happen, and it does really happen. So moving on to somewhat, but not completely, less complicated situations, we have 18q minus, in which someone is missing a piece of any size from any region in the long arm of chromosome 18. And these collectively occur in about 1 in 55,000 births. But again, there's some variation. On chromosome 18q, there's one region right here, has 2 genes in it, and it's never been found deleted in anyone. So what that does, it creates 2 groups. There's proximal 18q, proximal meaning proximal or closer to the centromere. And that group of individuals includes about 17% of all the people with an 18q deletion. So it's a smaller group. And there are 87 genes in that region. No 2 people have exactly the same deletion, and no one person has all 87 genes deleted. Then there's distal 18q minus, distal meaning further from the centromere. And that is the largest group of people with 18q deletions, 83%, and there are 102 genes in that region. But again, there's almost no one missing that entire region. And to make it a little more complicated, or to make it more clarifying on the terms we use, Someone who has a deletion from the end of 18 is said to have a terminal deletion. In this case, it's a distal terminal deletion. But they can also have an interstitial deletion of the same distal region, but the end of the chromosome is intact, and it's just this middle piece right here that's missing. So that's called an interstitial deletion. And they still have distal 18q minus, but it's interstitial. However, someone with proximal 18q minus only has an interstitial deletion because it only involves this interstitial region of the chromosome. So you'll hear the terms distal and proximal and terminal and interstitial, and just know that proximal 18q minus all have interstitial deletions, but distal 18q minus can have a terminal deletion. Not that it's a terminal condition, it's the terminal end of the chromosome, or an interstitial deletion. So a little bit more simple is 18p minus, individuals that have a deletion, again, of any size of any region of the p arm, and it occurs in about 1 in 56,000 births. And this is a little less complicated in the fact that about half of these individuals have a deletion that includes the entire p arm, and the other half of the group have a smaller deletion, every single person with a different breakpoint. So they're all unique. Or half of the group are completely unique. Then there's ring 18, in which they have one normal copy of 18, like all these individuals, and one copy forms a ring. And that is much more rare, occurring in 1 in 300,000 births. And by definition, in order to have a ring chromosome form, both of the ends, the caps called telomeres on either end of the chromosome, have to be missing. I don't know if it's true, but I think about it as the caps are gone and the ends are sticky and it forms a ring. And what happens is, by definition, there's missing genetic material at the end. Sometimes it includes genes and sometimes it doesn't. And every single person has a different amount of genetic material missing in total from either end of the chromosome. I think there's one person who has no genes missing from the Q arm and another with no genes missing from the P arm. And most of them have a combination. So some of them, as far as the families are concerned, will feel like they fit more with the 18q families or the 18p families or the ring families. So again, lots of variability. And even less variability, but more rare, is tetrasomy 18p. And that occurs in 1 in 625,000 births. And that occurs when the individual has an extra chromosome, but it's made up of 1, 2, 3, 4 p arms, so tetrasomy 18p. And almost everyone has exactly the same genetic change. We've had a couple people in the study who had a little bit of the Q arm in their duplication, but that was very, very rare, and they were had much more serious health consequences. So most of these individuals are the same. But that just goes to show you, as I keep saying, anything can happen, and it usually does. So again, these are the chromosome 18 conditions. As I said, the registry groups all these conditions or all these individuals into these 5 groups. But there's a lot of variability, and I think people should know that if you have ring 18, for example, and your child seems more like the kids with 18p minus, feel free to join in the 18p minus group. They're good with that. Don't worry about it. This is all about finding your community and finding others who are experiencing a similar thing. And so why do we do this? Here's an example. This individual has a distal interstitial deletion of 18q. And what we've been able to do since we started, when we started, this is all you could do is have this drawing. You could look under a microscope. That was it. Now, because the human genome has been sequenced, now we know exactly which genes are in that region. So that's very exciting. What we're working on, and we've made a lot of progress on toward, is possible consequences of each of those genes. Many of those consequences are none. Others, we are learning to define more carefully. And for some of them, we can advise typical therapies, whether it's a pharmaceutical therapy or a dietary change or just a physical occupational therapy, all kinds of therapies. This is what we're working toward. Most importantly, though, if this were the case, we would also be able to tell you these genes, don't worry about them. They're not included. So in a giant list of everything this person might see on distal interstitial 18q, once you know which genes and we are able to know what each gene does, you'll know what applies to you and what doesn't. So that's where we're trying to go with this, and we've made a ton of progress. And you can learn more, of course, on the registry website. Our syndrome descriptions really based or focused on new families coming into this, learning all the terminology. And there's introductory videos for each one and all kinds of videos on the registry website. On the Research Center website, which you can get to through the registry website, are all the management guides for all of these conditions, and the way they are organized. I have no idea what time it is. Let's see. The way they're organized is the first page is what we call a 60-second summary. Something you might want to give your child's ophthalmologist. Send them before they see him. If they see tetrasomy 18p, they're going to think trisomy 18. Believe me, I know the words are different, but the mind just does that. And so you want to give them something that's going to let them know they don't need to know about all the genes on 18. They need to know this is going to be an ambulatory child. And in fact, they're probably going to be a very active child and maybe even have something to say to you about all this. So not trisomy 18. And so it's just a one-page, real quick summary to give them an idea of what it is, so they have concept in their mind. The second page is what I think of as a table of contents, just kind of a very brief one-page summary of what to do when they're diagnosed, what to do in the newborn nursery, what to manage, monitor as they get older. And the next, I think for 18q is like 35 pages. Are all the rest of the information and all the developmental information that we have. This is still not good enough. It's really amazing. No chromosome abnormality has all this stuff, but this isn't good enough. For example, let's see, say proximal 18q, for example, is presented as if they're all the same and they're all different. And so we're working toward updating these and making these much more specific for individuals. And most importantly, thank you to the Chromosome 18 Registry and Research Society for supporting this work. It would not be done were it not for the donations from the members of the registry. That's really what supports this. There's no magic foundation out there supporting this. The National Institutes of Health will support a little fraction here or a little fraction there for a very specific project. But almost all of our support has come from the Chromosome 18 Registry membership over the years. So please remember that and give accordingly. And if you have questions about the research, please contact Catherine McCarthy. She's our participant coordinator. Here's her email address and her phone number and our website. So if you have any questions, and I have no idea, here's the schedule. When was I supposed to quit? Oh, I'm not too bad. Okay, only a little bit over. So are there questions or
Hi,
I
my
can't
name
see the
is
chat. So,
my name's Kirsten. I'm 32. I live in Oklahoma. I got diagnosed 2 weeks ago. I've been like trying to find an answer since we were little, like since I was little with my parents. They didn't tell, like, I'm still seeing my geneticist and they are rerunning my Gene DX. I sent it to Jessica through her email. I contacted you guys like last week.
okay.
And they asked me to swab my parents who are still alive. I swabbed them and we sent that out. They diagnosed me, they said multi-gene deletion syndrome, but it was 18q12.1. They said I have about 30% of that big black area on the 12 like missing. They said it
Yeah.
was like 16 total deletions that they saw, 6 were complication causing. But some of the things I don't have that the other people, I'm also, I guess, a bit older than some of the people like
No,
that.
probably not.
Okay, that's good. Like, because everything that I've been like looking at, it seems like children, like people are finding it
Yeah.
with their
Well-
like their children. And I have 2 children. After they told me I couldn't get pregnant, and then I was able to get pregnant. And then after I had my daughter, my son started following my medical pattern is when I started actively like trying to find an answer because I just kept getting diagnosed with different things growing up. They said like it was like ADHD and dyslexia. And then they started to notice that I had like bone shifting in my chest and I didn't get diagnosed with skeletal dysplasia till 2023. But now
So,
I don't even know if
yeah,
that's
so
a
it's
thing.
nice to meet you, Kristen. This is going to be a long conversation that we don't have time
Yeah.
for today.
Yeah.
But one of our sort of overall issues, I guess, is that parents are motivated when their child is diagnosed, usually at a young age. And so they're motivated then to join, find other families, get information. And so our cohort really started very young. We're getting to have more adults because those who started out young with us 30 years ago are now growing up. So we have fewer adults. My guess is there's many more adults like you are pretty mildly affected um, in the grand scheme of things and made are just now finding out. So I'm hoping a situation like yours is becoming more common. But I will have to have a conversation offline about your particular case, if that's okay, because it sounds very complicated and interesting. Well, everybody's case is complicated.
Yeah.
And so
Spectrum for a reason.
yeah, but thank you. And so stay on the call or on the webinar for the rest of the day. And so I look forward to Catherine and Brittany. Well, I see Brittany's on the call. Wave Brittany. Brittany is one of our research assistants. You may hear from Brittany as well. Um, so we'll be getting in touch with you this week then if you've sent us something. Okay.
Yes,
All
ma'am.
right. Thank you, Kristen.
You too.
And Jannine, I can field questions for you from the comments and from any of the attendees. But I thought Kristen asked, or what she asked led to a great question for families that might be newer. Can they ask specific questions about their diagnosis or their child's diagnosis to the CRC team or the team at the Clinical Research Center?
Yeah, and actually, sort of part of the next talk, but that's okay. Absolutely. I mean, you don't have to be enrolled for us to answer a question. If you are enrolled, usually we can give you a better answer because we know more about you because we've got medical records and et cetera. But yeah, we're happy to try to interpret a genetics report or whatever. You don't have to enroll to talk to us. We're happy to talk to anybody.
Thanks, Jannine. And just wanted to check on our schedule. Did you have a part 2 that you were going to do for the research
Yes.
update? Yes.
Yes.
If you want, you're welcome to move into part 2 and we can take questions after, or would you like to
Okay.
open up the floor for a few more questions now?
I don't, anybody have a burning question? Okay.
I'm not seeing any, so if you want to move on to part 2, that's no problem, and then we can do the Q&A after.
Okay, that works. Okay, so part 2 is really an introduction to the Chromosome 18 Clinical Research Center and what we do and our strategy for treatment development. So first of all, the Chromosome 18 Registry has a scientific medical advisory board. Many of these people, let's see, you're going to hear from some today. Many are parents or people, hopefully you've met all these people if you've been to a conference or two, to help think about what the biggest needs are for research going forward with registry funding. Unfortunately, they haven't had much to do lately because registry funding has been at a limited level and it's only allowing us to do the basics, which I'll talk about. Here's where we are, which I realized when I looked at this earlier that this is a pretty old picture because now there's this giant new Women's and Children's Hospital over here that's like this big and will take up half the picture. But I also noticed there's my car right there. So this is UT Health. This is where we live on the 5th floor in the Department of Pediatrics. And our goal is to make the chromosome 18 conditions the first treatable chromosome abnormalities. So what do we mean by that? We're really focused on self-sufficiency, which of course involves good health, but also independence. And we want to say no to SSI and trust funds, special needs trust funds. We'd love to have the day when no family has the need to deal with either of those things. And we get there through established therapies, making sure families are aware of and are able to get established therapies. And when there's not an established therapy, we plan to develop, or we are going to develop novel therapies. So if you come to visit us up on the 5th floor in the Department of Pediatrics in the School of Medicine, you'll see these bulletin boards. Here's 4 of the 5. Annis, our previous syndrome coordinator, it's not syndrome participant coordinator, asked families to send her pictures that she could put on the bulletin boards. There's 5 bulletin boards. All these families, all these smiling faces, everybody knows where they are when they come down the hall and are anywhere near our office suites and labs. Our staff include Catherine McCarthy, who's the participant coordinator. We try to funnel all correspondence through Catherine, so she keeps track of it. If it's a question she can answer, she will. If it's something Brittany can answer, who's a research assistant, she'll send it to Brittany. If it's something for me or whichever doctor needs to answer it, she'll send it to them. More importantly, she will bug them until they answer you. And she knows that's part of her job. And sometimes, these people are really busy and it takes a little bit of bugging to get them to answer you. And Brittany Antopia, who's on the call, is a research assistant. She abstracts medical records and does database searches and all things research oriented, I guess. Amukta runs the lab and is also a research assistant in the office doing data analysis. So these 3 highly talented, let's see, all 3 with master's degrees, are dedicated to chromosome 18 conditions full time. And I think everybody knows Dr. Hale, who's trying desperately to retire officially from his job, but we're not going to let him go. He's always going to be a part of the chromosome 18 family. But on the official side, Dr. Camille Karkar, who's a neurologist, is our new medical director. And Dr. Gelfond is our associate director. And we support 10% of their salaries, but I can assure you they you offered have much more than 10% effort that they give to this project. So there are sort of part-timers, but invaluable and very dedicated to this. And then There's a whole bunch of people here who we call on, usually on a volunteer basis, for help, insight, depending on the project, and depending on the question. And as you can see, they're in all kinds of, psychiatry, neurology, endocrinology, physical therapy, sleep medicine, all kinds of medical specialties. And on the basic science side, stem cell editing, mouse models, et cetera. So we have a really great team of people we can call on for specific projects. So this is Team 18. So if you see any of these people at the conference and they have the little blue thing sticker on their badge that says Team 18, they're part of this group. So far, well, actually, this is as of the conference this summer, our enrollment numbers are over 700 families enrolled, and here are the numbers for each condition. You can see here we split out P plus and Q plus down here, and don't lump them in with trisomy 18. And they come from all over the world, mostly North America and mostly the US. But we do enroll families from anywhere in the world. Our problem is twofold in enrolling more families not in the US. One is our consent forms and our developmental surveys, they could easily be translated into any language. However, many of the developmental surveys we use are proprietary. They're only normed for English speakers. So we could translate them, send them to anybody around the world, but we can't use that data. So why pester them to fill out these forms when we really can't use the data to publish it? So that's one limitation. The other limitation is we need a blood sample. And we need to get that within 3 days because we set up a cell line. And so, depending on the export regulations in each country, that depend, and the logistics of FedEx and DHL and all those sort of overnight carriers really dictates which overseas samples we can get. As you see, we got 22 from Australia. It can be done. But Mexico is a problem. They won't let us ship from Mexico. So those families come over to Texas and find a way to donate blood. So it greatly impacts the distribution. This has nothing to do with the frequency of these conditions in all these countries. It has to do with language and just the logistics of getting a blood sample to us. And so enrollment, we say we have, we call this our primary enrollment, and individuals can enroll wherever they get the information, through our website, through the Chromosome 18 registry, or a physician referral. Physicians don't sign up families, researchers don't sign up families, families sign up families, because this is a lifetime longitudinal study. And our primary enrollment, as I've already mentioned, involves consent forms that will explain everything that goes on. If you have questions, you talk it through with Catherine. If she can't answer them, she'll find someone who can, just so you feel like you know and understand everything that's in the consent forms. We need medical records. Principally, we need the genetics report, because every once in a while, we get a genetics report that from someone who does not have a chromosome 18 abnormality, but there's the number 18 somewhere in it, and whoever told the family didn't understand how to read it, and so they thought they had a chromosome 18 condition when they didn't. So we want to make sure that they in fact have that condition that's related to chromosome 18. And then, as I said, a blood sample. So what happens next is everybody gets medical and developmental history surveys to fill out. And you'll get one at the beginning. And if you have, let's say, a 2-year-old who's enrolled, when they get to be, I don't remember all the neuropsych test ages. When they get to be 3, they might qualify for a different developmental survey. And when they're 10, it might be another one. So depending on the age, and in the adults, we ask every year or two how they're doing. Are they living independently or in a group home, that sort of thing. So those kind of surveys, everybody gets, no getting out of that. The other things are often very specific for specific groups. That's why these arrows are different sized arrows. And what I should have put on here is, which I meant to do and failed to do. For example, we have a video assessment happening right now of individuals with 18p deletions looking at speech and facial movement to look for early signs of two different conditions. So that's specifically 18p minus families only. And it's a video that they do on their cell phone following a specific protocol. And anyway, Catherine can set people up for that. But sometimes we'll do an autism survey or a seizure survey. We are doing a seizure survey right now. I think we have forgotten what percent we have back. I'm really bummed. I think it's only about 70% of people have sent it back, which is really bad. I mean, I want to see 100%, especially from people whose child has never had a seizure. It's so important to know. We can't say what the frequency of seizures are unless we know how many never had a seizure. So it's one click is all that takes for people whose kids have never had a seizure. And sometimes it's complicated. And it's hard to answer all the questions, but please send back the seizure survey. I think in the next few days, I think we're going to close it because I was supposed to make a pitch for filling out the seizure survey right now. So your deadline is approaching fast on the seizure survey. Clinical assessments in San Antonio, those are very, very specific and only when we have funding to do a specific clinical assessment. We recently submitted a grant to the National Institutes of Health to allow us to do a specific clinical assessment. Keep your fingers crossed in January when it gets scored, and in May when they decide if they're going to fund it or not. And if it would start, it would probably be July. So when we turn that in the 1st of October, so that's the funding cycle. It takes forever. But NIH is not funding a very high percentage of grants. And so we got to keep your fingers crossed, light a candle, say a prayer, whatever it is you do to send good vibes our way. Please do it. So clinical assessments are quite limited and very specific. But then we have lots of collaborators, actually all over the world, who have specific skill sets. They might do genome sequencing or stem cell generation or gene expression. Those people are not part of our group. Excuse me. And that's why there's a red line here. They only get de-identified samples. They will never have your name. They will not have your address or your email or any way to contact you. All these collaborators who help us with all these things have no way of contacting you. So you should never be contacted by any of them. If you are contacted by a researcher wanting you to get involved in a research project that's not through us, give them our name. Maybe we can help them out. I mean, we do have almost, more than 700 people enrolled. So if they have a good idea, we're quite willing. And that's where a lot of our collaborations actually have come from, is people approaching us. And so, our goal is to get as much information as possible. So if somebody else has funding and a good idea, both are required, we're very excited to be able to collaborate with them. So that's sort of what we do and what we're all about in big picture. Our core activities that we focus on all the time, thanks to funding from the Chromosome 18 registry, is, of course, from that blood sample, we isolate DNA and set up a cell line. We collect medical records on an ongoing basis, which is why it doesn't work to have a doctor sign up a family, because the family needs to be committed themselves, because we're going to ask for medical records on an ongoing basis. As I've already talked about, we do surveys, we do DNA analysis, clinical evaluations when possible, and information for families, and that is the key. That's the whole purpose of all of this. The purpose is not to give a talk at a scientific meeting. The purpose is not to write a paper. Those are necessary evils, but that's not the point. The point is to get out information for families that are going to make a difference in people's lives. So why would you want to go to all this trouble? Because it is trouble to be involved in the research. Because as I said, we ask you to do all these things over time. I don't think I've ever heard anybody say we bug them too much. But why would you want to do it? Well, first of all, it's access to the team. For example, a week or so ago, we got a question from someone that sort of a typical kind of question. eh, but it was kind of, should we do? My daughter's been doing XYZ. Well, they were enrolled in the research study. So Brittany looks them up, tells us what condition this person had, gave us some history, and then that information could be added to the email the family sent and sent to some Dr. Karkar or whoever to answer the question, and they had background information. So they were able to come back with a more attuned and informed answer to that family. And if a new concern comes up, we have a medical history we can refer to. And we can also look in our database and see if anybody else has it. Even though it's a new concern to you, it may be something we already knew about. Or one or two families had it and it hadn't sent up a red flag because it was only one or two families. So we want to hear about those things. And access to, the other part of new concerns, almost forgot. If we have a new concern, if you're involved in the research, you're going to know about it. For example, many years ago, and I see Marie on the call, Marie said, maybe I shouldn't call out people by name. I don't think she cares. You okay, Marie? Said, some of the adults would touch
I'm
us on-
fine, Jannine.
Okay, I knew you would be, Marie. I know
Go
I knew
for
you would be. Said some of the adults with tetrasomy 18p have low bone mineral density. What's up with that? We said, we never heard of that. So we sent out a survey to all the families that have you had this diagnosis. And that's clue number one to all those families. We asked the question because we had a reason to ask the question. Okay, so heads up. A few families answered. We sent that out to all those families again, saying this is what we have in our database and this is what the answers we got. We might suggest you go to your doctor and get an evaluation. Few people were able to talk their doctor into it. We got more data. Then we sent out a letter to doctors, said, "Dear doctor," for families to give to their doctors, a little more push to get evaluation. More families got evaluated. And we wrote a paper about it. But the distance between when that first question went out to families and when the paper got published in the literature was, I should look it up, is probably 3 or 4 years. And after the paper was published, maybe a year after that, I get an email from a parent saying, my daughter just had some sort of orthopedic surgery, which was not successful because the surgeon said their bones were mush. Well, what I sent them the paper. What can I say? Had they been in the research, or even if they'd been a member of the registry, they would have known that their child needed to take extra calcium and vitamin D. And maybe that surgery would not have failed. So there's reasons to be informed and be part of the community. Being part of the research, you'll learn faster. Being part of the registry, you'll learn before it gets published. When we did a previous Caesar survey, I did a little video on the results before it got sent off for publication. I probably technically shouldn't do that, but we're really all about information for families. So that's how you all will learn the latest results. So that's why to be involved. So our 3 main concerns are, of course, health, improving independence, and understanding and hopefully heading off aging things. Because as I already said, in response to the question in the previous talk, our cohort is kind of young, and we don't know what's going to happen as they age. So here's how I think about how we approach this. So here's sort of the biology of disease. Okay, so if we start with looking at the individual and trying to describe what's going on with them, and we realize, oh, they have a digestive system problem, and we investigate further, and then we figure out, oh, here's the organ, in this case the stomach, that's got a problem, and investigate further to find out this is the tissue and these are the cells involved that have the problem, that are creating this problem for this child, and then what genes instigate that. So that's one way to begin to understand these concerns, is by looking at the individual. But we sort of have an advantage in that all these individuals have a chromosome 18 abnormality. We know, in part, what their genetics are. We can think about, okay, we know these genes on chromosome 18 are involved in, I don't know, a stomach muscle issue, and we can begin to understand what's going on by taking the other route, understanding the genetics, which cells are involved, what's wrong with them, how does that affect the organ, and then how can that help the child if you can do something about it. So we have this sort of double-pronged approach, all focusing on how to help individuals with chromosome 18 abnormalities. And as I said, the three main concerns are health, independence, and aging. And the approach is, I think, as I said in the other talk, to identify established therapies, already approved sort of therapies that may make a difference because we understand the underlying origin of the problem. Or when there's not such a therapy, invent novel therapies. So when focusing on the individual and taking that approach first, how do we learn? We learn from you. We learn, families give us information, goes into our database, then we create information back to families. That's how we get information, and that's what it's all about. It's all about sharing with each other. And all that information has gone into creating these physician management guides, which I have no idea if new people have joined us. I can't see on my screen if our numbers have changed. So just to repeat what I said before, the management guides have a cover sheet that's very brief, just explaining the briefest outline of what the condition is, because the person you may have told the clinic they have proximal interstitial 18q, and they have no idea what that is. And so this at least give them a visual of what that is. That's the first page. For a physician you're going to see regularly, or is going to be more involved with your child or your loved one with the chromosome 18 abnormality, the next page is like a table of contents. And it just is an overview of the main things the main assessments to do and referrals to ask for, and the things to monitor as they get older. And the next 30 or so pages are all the data that support all that, and all the developmental and neuropsych data that we've accumulated over time. And over time, things have changed at the research center. We used to have this hallway all lined with filing cabinets and all these files. Eventually, those got scanned, and they're all the files, all the paper, and all the faxes that we get all get scanned and go into the server. And a good part of that went then into Access database or an Excel spreadsheet. Bridget, who scores all the neuropsych data, had all her data in Excel. Patty in the lab had her data in Excel. On the clinical side, a lot of it was in Access. And a few years ago, thanks to the development of a new specific clinical research specific database developed out of Vanderbilt, called RedCap. We are migrating everything to RedCap, which is the Access and Excel information. That's done. All the scanned records in the server are an ongoing process to make sure that it really is all abstracted and entered into RedCap. So that's an ongoing process, but it's going to revolutionize what we can do for families and the kind of information we can generate. So Dr. Gelfand is going to talk more about this, so I don't want to steal his thunder, but we're investigating machine learning abstraction, which probably Brittany will be jumping up and down, screaming with joy when that actually happens, that it can be done by machine instead of her reading all of those and abstracting them by hand. It will be a lot faster. But it also will allow us to get better reporting back to you in a few strokes of the keys, to create a medical summary and research findings that are specific for families. So on the genetic side, taking the genetics first approach, chromosome 18 has 263 genes. And if you look it up, you might see a little bit of variation, 264, 262, because most genes were identified on a theoretical basis. It looks like a gene to me, according to some computer program. This is my number, and I'm sticking with it because I had a kind of a high standard for what I called a gene. The 18P region has 67 genes. The proximal 18Q region has 87 genes, and the distal 18Q region has 102 genes. Okay, for you math whizzes out there, you're going to say, that's not 263. That's because there are 2 genes right here. Nobody has a deletion that includes those, and there are 3 genes up here, and no one's ever been reported in our cohort or in the literature with a deletion of those genes. So that's why 5 are missing. So, but good news is, and we started out maybe 25 years ago with the hypothesis based on fruit flies, okay? Model organisms can tell us a lot. Because of what we knew from fruit flies and the genetics of fruit flies, we hypothesized that really only 10% of these were going to cause a problem when you had a gene dosage abnormality. So dosage is the term we use for a deletion or a duplication. You have an abnormal dosage of the number of genes. And in fact, that's sort of turning out to be true. It's actually very much turning out to be true. Most of the genes are just bystanders. There's no dosage effect. They're okay. Your body, the cells, the organs, all, they work fine when there's 1 copy or 2 copies or 3 copies or 4 copies. It just doesn't matter. But we've got to figure out which are which are the culprits, the 10% that aren't bystanders. So one of the ways we do that is really focusing on eliminating some of them from concern. That's because all of us, you, me, everyone here, has deletions and duplications in various parts of their chromosomes. Those are all very small, mostly don't even involve a gene, but some of them do involve genes. And all these people think they're perfectly normal. I mean, who doesn't? And all the genes that have been found in these people who think they're normal, we can eliminate as part of our group of bystanders. We're not going to worry about them. We can also get information from animal models. There is a repository of mice that have a deletion of pretty much every single gene. Unfortunately, most of the investigators study the mouse when the mouse is missing both copies of a specific gene, not 1 copy, but sometimes we learn something about mice with missing both copies and they're perfectly normal. I don't have to worry about that gene anymore. And sometimes they have an issue that's related to something that we see in someone with an 18p deletion, for example. So we put that on our list of ones that we think are dosage sensitive. There are fewer mouse models for duplications of genes, but sometimes they're informative too. And we also use information from people, from single gene disorders. So I think everyone's heard of recessive conditions in which someone inherits a gene that doesn't function from mom and another one of dad's copies doesn't function. They have two copies. In each case, only one works, one has a mutation. And 1/4 of the kids on average will inherit both of those copies with a mutation and be affected. Cystic fibrosis is one a lot of people know about. But the point is, each of these parents have one non-functional copy. That's equivalent to having a deletion, and they're perfectly healthy. And so genes that are involved in recessive conditions, I don't have to worry about. It's not going to be dosage sensitive or quite unlikely to be dosage sensitive. On the other hand, genes that cause a condition because one copy causes a disease when there's a mutation that makes it non-functional or equivalent to being deleted, then that really is a dosage sensitive gene, and we want to know about that. So not every day, 5 days a week, I get updates on all 263 genes on chromosome 18, anything new that's been published on any of those genes in the week. And doesn't matter if I'm on vacation or it's Thanksgiving, I still get my updates every single day. And then there's a whole bunch of keywords that I get updates for. So this is a daily ongoing process of gene profiling to try to figure out which are the dosage sensitive genes, the culprits, and which are the bystanders. And this is what we have today. I don't want to spend a whole lot of time explaining it because it is about to go away with a whole new exciting, user-friendly, hopefully, version of this appearing very shortly. And Dr. Gelfand's going to talk about that, so I want to set him up for that. But a point I really do want to make in this is that you'll notice on the left are individual genes with the phenotype or characteristic that the gene is responsible for. And over here on the right-hand side, we don't know the gene, but we have it narrowed to a region. So these are the regions for a phenotype or a characteristic. Now, the important thing I want you to note is this one's bolded, this one isn't, this one isn't. So some are bolded, some are not. Over here, some are dark gray, most are light gray. The reason for that is the bolded and the bolded and the dark gray are those conditions that occur in more than 50% of the people with a deletion of that region or gene, whichever. There's a lot that aren't bolded and a lot that are light gray. So many of these only cause a problem in less than 50% of the people with a deletion of that gene. So that's turning out to be one of the common features of chromosome abnormalities is that many, even once you know the gene, the chance of having a problem due to that gene might be 20%. So it's at least we know something and we know what to watch for, but it's not a given. So right now for the deletions, there are of the 263 genes, 219 of them either have no effect when you have a deletion of one copy, or it requires a mutation in the other copy or some other gene in the genome before you have an effect. So that's 219 out of the 263. Only 13% cause a problem when in less than 50% of the people who have a deletion of that gene. And 14 of the 263 genes cause a problem in greater than 50%. I think only one gene, TCF4, which causes Pitt-Hopkins syndrome, causes a problem in 100%. And then there's 17 genes we still don't know anything about. So that's not very many. Some of these may turn out to not be real genes. They might be pseudogenes or something else that just kind of looks like a gene and it doesn't really do anything. So stay tuned for that. now. And what that does, like I talked about in the other talk, if you know your region of deletion, you can look up the information about each of these genes in each of these phenotype regions to know what you're most likely to have as possible issues. And it also tells you, don't worry about any of these things down here or these up here. This is what you want to focus on. This really does begin to guide in an intelligent sort of way what sort of things to watch for, what medical issues to be evaluated. Oh, whoops, oh, that's just what I said. Okay, so unfortunately, the duplications aren't so well developed. That's really because there are fewer and fewer people with small duplications that we have enrolled. But we do have about, I think, 25 more individuals who are enrolled, who we are getting long read sequencing done on. And we will take those data. They have small duplications. We will take those data and compare them to the medical records. And so hopefully in the near future, this will be built out a little more and begin to look more like the duplications. So where are we going as far as novel therapies? Well, when you think about therapeutic gene regulation, is really how we think about it, we're not looking at trying to put back a whole chunk of chromosome. Why? Why try to replace 30 genes in an arm of a chromosome? First of all, it's not technically possible and not even close to being technically possible. But why do that when only 3 of them matter? So our approach is a little different to understand those genes and to think about it. If you have an 18q deletion, maybe there's only 5 or 4 that you need to upregulate. Or for 18p, only a few you need to upregulate. Ring 18 might be a few more, but it's not 263. It's just a few that we have to learn how to upregulate. And with regard to the duplications, likewise, we need to learn how to downregulate those genes. And upregulation and downregulation of genes is an area of medical research that is ongoing for all kinds of genetic conditions. We don't have to, and once we know the genes, we may not have to invent the strategy. It may exist. Many drugs upregulate and downregulate existing genes. So there may be a drug that does this already. There may be other strategies for doing this that are just now being invented. So this is our approach to understand specifically which are the genes that are causing the problem. And in your case, is that gene causing you a problem? Because maybe it's not in your case. Even though it's considered a dosage sensitive gene, it may not be causing a problem in you. And so we learn the genes, figure out what evaluations to do, which genes really cause the problem, and then how do we affect those specific genes. So all of that is a known pathway for improving health in people. We just don't know the specifics for chromosome 18 abnormalities, and that's what we're working on. So thanks to the Chromosome 18 Registry and Research Society for sponsoring almost all of this work. And they support the work thanks to donations from members. And so you all who are watching are the people we need to thank. Pat your neighbor on the back and thank them for their donation, because that's what's made all this possible over the years. And if you have questions, the participant coordinator is Catherine McCarthy. Here is her email address, her phone number, and our website. Any of those methods work just fine for getting in touch with us. And because I'm in display mode, I have not a clue what time it is. Oh,
Jannine, we have about 10 minutes.
I'm pretty good. I'm almost
You
on
did
time.
great.
Yay, I did it.
So we have about 10 minutes before lunch. So if anyone wants
Yeah.
to type in questions in the chat or come off mute and ask their questions, we have 10 minutes. And then after lunch, we're coming back for a Q&A with all the staff. So it'll be Dr. Cody, Dr. Karkar, and Dr. Gelfand.
Mm-hmm. And maybe Dr. Hale too, I think.
That would be great.
I think, yeah. So all 4 of us. So, any questions? Oh, I'm gonna unshare, stop sharing. Okay, now I can see the chat.
Perfect.
There is. So, hi
And well,
Shannon, good to see you.
Hi,
Hi.
Jannine. This is Liz. Liz Woodfield. Good to see you. Thank you so much for the
Hi.
talk. Hi. Sorry, I had to wait till I parked. I've been listening to you as I've been driving children around. But is there research being done with other genetic conditions that we can learn from or steal from some of their advances?
Oh, absolutely. And we collaborate with those people whenever there are, and whenever I can discover them. Because for me, it means let them pay for the work. We'll collaborate with them, help them however we can, and then we'll turn that information into information for families. Because most research teams really could care less about that aspect.
With the Down syndrome group, do they do much research?
Oh yeah.
Since most people heard of Down syndromes and it's a larger group.
Yeah. Oh yeah. And yeah, there's not much going on I mean, some of the basic science approaches are sort of helpful. But not in a specific sort of way. Because it's a whole chromosome duplication, their sort of mindset is in a different place entirely. So they're in some ways.
So they're not looking to upregulate or downregulate some of the more offending genes, or
yeah, maybe, but they haven't quite figured out what they are necessarily, and they're not yeah, so some of that work is going on. And the strategy, I mean, we can, if successful, we can begin to copy that strategy. Yeah. But that's not, doesn't seem to be their big emphasis. The big emphasis seems to be in more the direct clinical care improvement kind of aspect and how the genetics affects that, but not novel things, at least as far as I know. I try to keep track of it, but.
Okay, great. Thank you so much.
Thanks for listening, even in your car.
I'm listening. Jannine, I've got a question for you, and it may be easy to answer and it may not be. I don't know. But could you speak a little bit to the group here about, obviously fundraising for us is a key thing to continue to fund, but what are some of the key things that, like, I'm not sure if I'm going to say it right, the National Institute of Health, perhaps, that it takes for them to want to fund a study? Because it takes a little bit of a bigger picture.
Brain transplant for them.
Yeah, right.
So it's changing for good and for bad. It's changing in that I don't know, years ago, maybe 10 years ago, we turned in a grant, and one of the criticisms was that this was only about 18Q minus. Well, duh, of course it was. That was the point. But it wasn't important. They only wanted to learn about fundamental mechanisms of biology. And if you had a condition name in it, they didn't want to, look at it. And if we picked out a condition, let's say, I mean, I don't know, autism or whatever, then we had to be part of the autism club, which we weren't. You can have a neuropsychologist, but they're not anyway, it's a click thing. One of the good things that's really happened in the last 10 years is they started the NCATS. What's NCATS stand for? National Center for Translational Science or something like that. That's disease focused. And so because of that, they are having, when you write a grant, you can just write a grant to the NIH. You can do that. your chance of getting funded is pretty small. But they also have, and they come out once a week. I get a list of them every week, all their funding opportunities. And they had a funding opportunity recently for, it's right here. Let me read it. Clinical trial readiness for rare diseases, disorders, and syndromes. Yes, something, yeah, it got rare disease right in the title. So they're not going to be, hopefully that won't kill us. So we have to depend on them to have something focused, and that is just so incredibly rare. And so we look for those all the time and apply for those because over the years, and all the many zillion grants we've put in, if you don't go with something that's really focused and fits what they're looking for, it's a completely wasted effort, and it's a month of work that was wasted. I mean, I could write grants all the time, but it would be wasted most of the time. When I was in graduate school, the funding, what they call the pay line, the funding, they funded about 30% of grants that were written. So you kind of thought, okay, I'll write 3, I'll get 1. Okay, 3 months, I get 1, I'm good for 5 years, I'm okay. Now it's like 10%. So if you think about, you can't write 10 to get 1. That's 9 months wasted. I mean, it is, here, wait, let me show you. Where's the grant? Got a grant. Here we go. This is a grant. Takes a little while to do it. And it takes a lot of people to get them coordinated. Anyway, so it's not feasible unless there's a really targeted funding opportunity, and we're always looking for them. And that's why we really have had very little funding from the National Institutes of Health. We were lucky enough to be included at some level. I don't know why, but in the Gabriella Miller Kids First Pediatric Research Program, which was looking at structural birth defects. And so they were enrolling cohorts from around the country that were already established cohorts with specific birth defects. So one group had conductive hearing loss, another had congenital heart disease. I said, well, we've got, 20 different birth defects. And they thought, that sounds really interesting. Maybe you should apply. So, okay, when someone tells me maybe I should apply, I apply. Even though it was a little weird, but in a way, we know some genetics already. We have a given chromosome abnormality. So our version of a congenital heart defect has one aspect known already. Maybe we don't know which gene, but we've got to narrow down. And so it can help the other groups in that way. And so we did get those grants, and we have one right now from them. And we also have a grant that is not supporting, how to explain this one, supporting our basic science collaborators. It's through us. So we have like 4% of it, but it's supporting collaborators to make cerebral organoids and mouse models for specific, for dysmyelination.
And thanks,
So
Jannine.
it
Sorry
can
to
help us. Oh, okay.
Oh, go ahead. I'm sorry to interrupt.
Sorry.
This is such a great thread, but I just wanted to let you know it's about that time to break for lunch, but then we're coming back with the Q&A. Did you have any closing statements about that question?
Oh, no, just thank you for participating. Thank you for being here. Thank you for donating to the registry. Thank you. It only happens because we're all in this together. And it really takes all of us to do whatever it is our individual gift is to support each other and get all this done.